Blood typing is a method to tell what type of blood you have. There are about 36 different blood typing. Blood typing are all about code of proteins or sugars on the surface of the erythrocytes. In this lecture, I will go over the most important classification system, that is ABO blood system, and second most important RH blood system. Before we will talk about different group types, you need to know some important terminology. First is what an erythrocyte. We just covered it in the lecture 21. Erythrocyte is a red blood cell. You know this already. You need to keep in mind that antibodies are made by plasma cells responding to a presence of antigen. Plasma cell is a type of immune cell that makes large amounts of specific antibodies. In short, it is an activated B cell. The purpose of this lecture is not to introduce you to a structure of an antibody protein, but you have to understand that shape of the antibodies are very specific. The diagram on the slide is a comical representation of antibodies. In fact, what you see and will see during the whole lecture is not the whole antibody, but a tiny portion of it that matches or does not matches an antigen on the surface of erythrocyte or erythrocytes. Similarly, it could be a match of a bacterial antigen, but about this I will talk during my other lectures. By binding to antigen, antibodies make it immovable and group cells to form clusters. Cells clump together. Remember, the antibody is a protein made by plasma cells in response to an antigen, a substance that causes the body to make specific immune response. Each antibody can bind only to one specific antigen. The purpose of this binding is to help destroy the antigen. I repeat again, antigen are any substance that causes the body to make an immune response against the substance. Often when people talk about antigens, they mean foreign particles that come from outside the body. It could be a toxin, a foreign chemical, virus, or bacterium. Something that do not belong to a body of a certain organism, including humans. In this lecture, we will refer to an antigen that is located on the surface of a red blood cell. Yes, antigen is a protein that is sitting on the surface of erythrocyte. Here is an antigens on the surface of an erythrocyte. I hope you understand that this is just a comical version of proteins on the surface. In reality, these proteins are very tiny. After all, they are molecules, and you will not be able to see them under the microscope. These surface molecules called antigens. In a few minutes, you will understand why we call them antigens. Here, I label both of them for you. You see an erythrocyte of a person who has type A blood. This blood type is determined by the A antigen, which is specific portion on the cell surface. It is like a flag. Look at me. I am not just an erythrocyte. I am type A erythrocyte. Not B, not AB or O, but type A. Type B blood has erythrocytes with different type of antigens on its surface. We call them B antigens. Type AB blood has erythrocytes with both types of antigens on its surface, type A and type B antigens. And type O has erythrocytes with neither A nor B antigens. None of them. These erythrocytes are naked or not so naked, as you will see later on. Now we want to take a look at the plasma of different blood groups. We just examined different types of erythrocytes. Now we want to examine the plasma, a liquid matrix of these types of blood. You see, in the group type A, plasma cells quickly produce B antibodies so they can destroy B antigen by binding to it. Yes, the plasma of type A blood filled with type B antibodies. Let me once again remind you what we are looking at. We're looking at erythrocytes and the matrix that we call plasma. We look at proteins called antigens. They are on the surface of erythrocytes. Since we are taking a look at the type A blood, we see the A antigen on the surface of the cell. And we see B antibodies since it is type A blood. Remember, plasma cells of type A blood produce B antibodies. And in the group type B, plasma cells produce A antibodies. So they can bind to A antigen and destroy it. Different type of blood could be deadly enemies. You can clearly see on the slide how specific the shapes of the antibodies are. Antibodies B shape on the way so they can match B antigens, and the antibodies A shaped to match A antigens. If antibodies matches the antigens, they will form clusters, or another way to say it, they clump. Or there is even a better way to say agglutination occurs. Agglutination is the clumping of particles. The word agglutination comes from the Latin agglutinare, gluing to. But people with type O can receive blood only from the donors with type O blood, because their plasma cells produce antibodies against all other blood types. Though they can donate their blood to anyone, since their erythrocytes do not have any antigens on their surface. They are naked. This means that if we transfuse type O blood to any other blood types, the erythrocytes will not trigger antibody production and clumping of the blood cells. Therefore, people with type O blood considered to be universal donors. Blood type AB does not produce any antibodies against another blood types. Therefore, the patients with this blood type are known as universal recipients, since they can receive blood from donors with any blood types. Afterwards, they do not produce antibodies against any of the blood types, so the clumping of blood will not occur. Their blood will not form antibodies against A, B, or O type, and certainly AB blood types individuals will not produce antibodies against its own antigens, which is A and B. If this is not clear, let's take a look at different types of blood transfusion scenarios again. Learning is a matter of repetitions. Let's say we have donor B and his blood we will transfuse to recipient with blood type A. What will happen? If you say clumping will occur, you are correct. Our recipient will die from this transfusion because clumping will occur. The cell become bound and immobilized, like Tantalus on the painting of Raphael Poggi. Let's say now we have donor A and we will transfuse his blood to recipient AB. Will the recipient produce antibodies against donor with type A blood? No, because type AB blood individual do not produce any antibodies against other blood types. He or she is a universal acceptor, so our patient will survive this transfusion. Now we will take blood from donor B and transfuse it to a recipient with blood type O. What will happen? The recipient with type O blood will die because it will produce antibodies against antigen B from the donor and clumping will occur. And now, by looking at the diagram in front of you, let's answer more questions. Which blood types can receive type A blood? Please pause the video before you are sure of your answer. And the answer is A and B. Why? Because person with blood type A will not produce antibodies against its own blood types. As you remember, and as you see from this diagram, type A blood individuals produce type B antibodies. Shape of type B antibodies does not match the shape of their antigen. Therefore, clumping does not occur. AB can indeed receive type A blood because type AB does not produce any antibodies against mentioned blood types. Now, another question. Which blood type can receive type B blood? Pause video to think about the question. And here is an answer. B and AB. The explanation to the answer is by analogy, the same as the answer on the first question. B can indeed get type B blood type, and it can get type AB blood because AB does not produce antibodies against A and B types. Let's move on further. Which blood types can receive type AB blood? Pause the video. The answer is AB only. Why? Because type AB blood has all two types of antigens sitting on its surface. So this will elicit an immune response in the recipient or acceptor. And if we will do AB transfusion to types A, B, and O, they all will die from the clumping of the proteins in their blood circulation. If you still lost, look at the shapes of antibodies and antigens. Clumping will be unavoidable. Those antigens like enemy flags hanging there and screaming out loud, kill me, kill me. Next question, which blood types can receive type O blood? Pause the video and think. The answer is A, B, AB, and O. Why is it so? Because erythrocytes of type O blood do not have neither A or B antigens on their surface. Therefore, none of those blood types will elicit immune response. There will be no clumping occurring, or we may say there will be no agglutination. The A and B antibodies have nothing to bind to. Therefore, type O blood is known as universal donor. However, type O blood can receive only type O. Why? Because it produces both type A and type B antibodies. Look at the diagram last drawing. Interestingly, indigenous people of Peru all have type O blood. Now we will go to another level of complexity because you can be either rhesus negative or rhesus positive. There is Rh factor. It is all about measuring for a D antigen. If you have D antigen, you are Rh positive or rhesus positive. If you do not have, you are rhesus negative. So let's again look at the erythrocyte antigens. Look how this slide is different from previous one. What do you see here? You see additional antigen, don't you? Let's compare this diagram with the diagram from previous slide. Rh factor is an inherited D protein found on the surface of erythrocytes. Rh factor named after rhesus monkey in which D protein was isolated. If your blood has the D protein, you are Rh positive. If you do not have D protein on the surface of your erythrocytes, you are Rh negative. So let's label the ABO groups accordingly. The Rh factor is usually indicated by a plus or minus sign shown after your blood type, such as A plus or AB minus. Presence or absence of D protein makes matching blood types more complicated. The Rh factor must be matched during the blood transfusions so that agglutination does not occur. Either A negative or A positive, or some of you can be either B negative or B positive. The types A, B, and O could be positive or negative as well. Rh factor is all about measuring for a D antigen. If you have D antigen, you are Rh positive. If you do not have the antigen, you are Rh negative. Am I repeating myself? I think I do, don't I? Now, keeping in mind what I just said, answer the question. What cells will produce antibodies against D antigen? Again, what cells will produce antibodies against D antigens? The answer is very simple. It is Rh negative cells or Rh positive cells. Here is the answer tip. Remember that antibodies are specialized immune proteins that are produced based on the antigens that are not present on your red blood cells. Pause the video and answer the question. What cells will produce antibodies against D antigen? The answer is all ABO blood types that are Rh negative will produce antibodies against D antigen. Obviously, Rh positive cells will not produce antibodies against their own antigen. You remember when a few minutes ago we came to the conclusion that it is fine to put A-type blood into AB recipient. But now, keeping in mind the antigen, you hopefully realized that things might not be the same. So please answer the question, what will happen if we take A rhesus positive blood type and transfuse it into AB rhesus negative? Will there be clumping or no clumping? Pause video if you have to. There will be clumping or agglutination. So A rhesus positive blood types should not be mixed with AB rhesus negative blood types. This is because AB rhesus negative recipient will form antibodies against the antigen of the donor. As you can see from the data table, most common blood type in the US is O positive, rhesus positive, and A rhesus positive. Generally, rhesus positive is much more common than rhesus negative. Having a rhesus negative blood type is not an illness, and it usually does not affect your health. But it can affect pregnancy. A pregnancy needs special care if mother is rhesus negative and her baby is rhesus positive. That's called rhesus incompatibility. A baby can inherit the rhesus factor from either parent. When the baby is rhesus positive and mother is rhesus negative, the mother is carrying baby with an antigen that she does not have. If during the childbirth some of the baby's blood enters the mother's bloodstream, the mother produces antibodies against the rhesus positive antigen. Once mother has these antibodies in her blood, her body uses them to attack rhesus positive antigens of future babies. This could cause the woman to miscarry her next baby. The condition is called erythroblastosis fetalis. Physicians are now able to prevent this condition from occurring. After the birth of rhesus positive baby, physicians inject the rhesus negative mother with substance that destroy any antibodies she has produced against rhesus positive antigen. This procedure ensures that the next baby the woman carries is protected. Now you can imagine why first blood transfusion were not successful. Physicians did not know enough about blood typing. Let's take a peek into a blood transfusion history. The first transfusion of blood in the modern sense is reputed to have been done at Rome in about 1490. Pope Innocent VIII lay down from the terrible disease called extreme old age. A physician attempted to rescue the pontiff by transfusing into his veins the blood from 3 small boys who were aged to receive money award. The testimony as to what actually transpired is conflicting. According to the one version, the blood was used to prepare a draft for the illustrious patient. There is no assurance that he received either the transfusion or the drink. All seem agreed, however, that 3 boys died, that the death of the Pope was not slayed, and that the physician fled quickly as possible after the failure of experiment. The actual history of blood transfusion begins with the discovery of the circulation in 1628. In 1628, British physician William Harvey discovered the circulation of blood. Electrified by Harvey's immortal contribution, investigators throughout the world begin to experiment energetically with the infusion of various substances into the bloodstream and to speculate upon the feasibility of transferring the blood from one arteries of one individual into the veins of another. It is in all probability that because of the direct stimulation of Harvey's lectures that the first coordinated and scientific studies of infusion and transfusion were made in England. The first known blood transfusion is attempted soon afterwards. In 1665, the first recorded successful blood transfusion occurs in England. Physician Richard Lower keeps dogs alive by transfusing blood from other dogs. 1818. British obstetrician James Blundell performs the first successful transfusion of human blood to a patient for the treatment of hemorrhage. In 1907, Rubin Ottenberg performs the first blood transfusion using blood typing and cross-matching. Around 40s, the RH or rhesus blood group system is discovered by Karl Landsteiner, Alexander Wiener, Philip Levine, and R.E. Stetson. The beginning of the modern era of blood transfusion coincided with World War II and the resultant need of massive blood replacement. In the right side of the slide, you see my grandmother in Irkut city who is getting ready to donate blood to the Russian soldiers fighting German Nazis in 1943. Nowadays, every 2 seconds in the US, someone needs blood transfusion. This blood comes from people who donate it. As Dr. Robert Bill put it, blood transfusion is like a marriage. It should not be entered upon lightly, unadvisedly, and wantonly, or more often than it's absolutely necessary.